Hi, my name is Angela Robinson, and I'm going to take you on a tour of the animal cell. The animal cell is a eukaryotic cell. A eukaryotic cell has membrane-bound organelles, linear chromosomes, and a nucleus. They are much larger than prokaryotic cells, which lack these characteristics. These small bound organelles allow compartmentalization for various functions to be separated, but their work is interdependent. Let's begin our tour. This is the endoplasmic reticulum. It is composed of sacs and tubules which modify lipids and proteins. The ER is composed of two compartments. The smooth ER is the small yellow figures on the pink structure that help synthesize lipids, carbohydrates, and detoxify substances like alcohol. The rough ER is the light pink membrane between the tubule sacs. It helps synthesize new proteins after translation by adding side chains. The nucleus is found in eukaryotic cells. This is where DNA is housed. In the nucleus lies the nucleolus, where RNA is transcribed like mRNAs and rRNAs. Inside the nucleus, DNA is safely housed in a complex. The complex is in an unwound packet called chromatin, or heterochromatin. It can't be transcribed. However, during transcription, the complex histones unravel euchromatin, which can be transcribed. When we zoom into the nucleus, we see small holes called nuclear pores. When modified mRNA leave the nucleus, they pass through the double membrane nuclear membrane envelope. The mitochondria is the final area where most of the ATP from oxidized glucose is catabolized when oxygen is present. It is a double membrane structure which increases surface area for electrons to power ATP synthase to generate ATP. The yellow figures are called lysosomes. They have low pH enzymes that can break down many macromolecules like proteins and lipids. The small yellow spherical organelles are called ribosomes. They synthesize proteins during translation when mRNA from the nucleus are read through the ribosome to code proteins. In the pancreas, we have a lot of ribosomes where insulin, a hormone, is secreted to lower blood glucose levels. The green spherical structures are called peroxisomes. They have enzymes that break fatty acids and amino acids. These enzymes safely break down peroxide into oxygen and water, which accounts for any oxygen radicals that could damage cells due to instability of electrons. The yellow rod organelles are called centrosomes. They are found only in animal cells. They pull replicated chromosomes in opposite directions to divide the cell. Near the ER is the Golgi apparatus. Proteins, lipids, and carbohydrates that have gone through the ER may be sent to the Golgi for further processing. These compounds enter through the cyst connected through the trans in or on vesicles to be expelled out the cell membrane. Cells are mobile and they have a certain structure called the cytoskeleton. Some cells have a flagellum or cilia which help to project the cell in different directions. Cilia are tiny projections while flagella are long tails. The cell is confined in a selectively permeable membrane called the plasma membrane. It is comprised of phospholipid bilayer, which allows certain molecules to pass. Its layers is made out of phospholipids, which are comprised of two fatty acid tails, which are hydrophobic, and a phosphate head that is hydrophilic. This structure is important because it regulates what can enter the cell. Large molecules like ions or charged particles can't enter the cell without the aid of protein channels. Well, that's the whole tour of the eukaryotic animal cell. Oops, we forgot about this ER. Well, as you can see, eukaryotic cells are very complex organisms. My name is Angela Robinson, and I hope you have enjoyed our video.